Hello, everybody. I would like to thank organizers for giving me opportunity to share the results of our research with you in ISMB ECCB conference. So let me present our research, which is entitled, "Is the success of AlphaFold due to a better understanding of physics?" Authored by me and Marina Pak. So we all know about tremendous success of AlphaFold, and here is AlphaFold performance in CASP 13 and CASP 14 experiments. And here you see that in 2020, AlphaFold version number 2 achieved the near to experimental accuracy of 90% of GTTS score. And after that, the problem of protein structure prediction was declared to be solved, and it was tremendous success to all science. And here is AlphaFold performance in CASP 14. AlphaFold is the deep learning algorithm, and it was trained on PDB structures published by 2018, uses multiple sequence alignment and PDB, and the RMSD, median RMSD, was less than angstrom. However, for all competitors, it was close to 3 angstroms. And from original paper, we see that the prediction of protein structure was beautiful, even for huge viral polyprotein. And the questions arise, what is the main reason for this success? What does AlphaFold actually do? Does it predict three-dimensional protein structure from the physics of protein chain, or does it restore the 3D structure using multiple sequence alignment and PDB statistics learned during training? And since AlphaFold is a deep learning algorithm, so we cannot ask AlphaFold directly. We cannot open the AlphaFold and easily see what is the protein potential function written there. Is it written there or not? And also the second note is that at some moment AlphaFold uses the relaxation to get the final structure, and it uses some force field, conventional force field. And we, of course, when we talk about this problem, about understanding by AlphaFold physics, we do not mean the physics that is inside the relaxation mode, because almost every tool uses it, and in respect to that tools, we do not tell that they understand physics to that extent. Actually, what we ask is, does AlphaFold understands physics, some extra physics, compared to other tools, which allows it to predict well? So actually, every tool and every human understands physics of proteins to some extent. For example, if we are given some protein sequence and we are sure that it is protein, and we are given 4 structures with high probability, we choose this structure because it is most probable, because those two are two unfolded, and here is the long alpha helix. And so, as I said, we will choose correct structure, and so we understand physics of proteins to some extent. However, when we ask the question about AlphaFold, we ask about some extra physics that allows AlphaFold to outperform other tools. And reasons to be skeptic about AlphaFold's understanding of physics was raised immediately, because from the original paper, we see this beautiful prediction of the side chains. However, in the native structure, they are coordinated by the zinc ion, and AlphaFold predicts those side chains in the absence of zinc ion. And another more dramatic example was given in the same paper. When AlphaFold was given one chain from a multimer, it predicted it well inside the trimer. However, it was given only one chain, and by default, it was asked to predict just monomer structure. And of course, this structure cannot be monomer structure. And today we will talk about ranking decoys as a potential tool to resolve this question: does AlphaFold understand physics or not? And we will talk about ranking distant decoys and ranking close structures. First of all, we will talk about ranking distant decoys. And ranking decoys, actually, it corresponds to one of the key ideas in protein structure prediction used in threading when we give a sequence to a threader, and it takes on different structures from PDB for sequence and estimate the energy of that sequence in that structure. And after that, all that variants are collected and ranked, and we see some plot from which we can conclude how good is the potential energy function used in the particular threader. And following our colleagues, actually before that we considered AlphaFold as a black box, and we cannot do anything inside it. However, following our colleagues, Roni and Ovchinnikov, we made the following. We repeated their setup. We prohibited AlphaFold to use multiple sequence alignment. We supplied empty multiple sequence alignment, and we also can prohibit AlphaFold to use library of templates, PDB. So we can use only one template. We can give only one template to AlphaFold, and so input can consist of only query sequence and decoy. And so in this case, AlphaFold acts as a threader. So its prediction of the query sequence will depend on decoy, and we can estimate the decoy performance in that setup. And for the datasets of the structure, we use the novel proteins released after AlphaFold and dissimilar to AlphaFold training set, and also novel folds from CASP14 targets. Here is the distribution of protein length of CASP14 targets and new novel fold structures. And as the dataset of decoys, we used native structure, of course, then long alpha helix with the length equal to that of the native structure, and also decoys generating using 3D ropes. It was stochastic process, so we see different number of decoy structures generated for different PDBs. And of course, after ranking, when we talk about ranking by AlphaFold of decoys, so for example, here was native structure was given as a decoy. Here some other structures were given as a decoy, and here is was alpha helix given as a decoy. And so when considering this ranking, we of course should compare to some other ranking tool, some threader, and as this we chose this threader to be the threader of ITASER2. ITASER2 is the best automatic tool in protein structure predictions since 26, so that we considered ITASER threader Muster as the best threader during last 15 years or so. And we see that when we look at the correlation between predicted TM score and TM score between native structure and decoy, we see that AlphaFold gives only 54% of Pearson correlation, and the Muster shows 69% of Pearson correlation between the score and TM score. And so when comparing metrics as is, Muster outperforms AlphaFold in decoys ranking. However, following our colleagues, we see that we used composite score. And why did we do this? Because we see here in the diagram those structures that are easy targets. So actually here we classified all the structures into easy, hard, and medium, and the prediction of easy targets does not depend. Did we allow AlphaFold to use MSA template library or not? The prediction was perfect in all cases, and those were easy targets. And for hard targets, the good prediction was possible only when we give decoy native structure as a decoy. And so if we color by the category of the targets, and we see that all those of this cloud, part of the cloud is consists of easy targets. And so we see that AlphaFold predicts easy targets well, even with no multiple sequence alignment and templates, and AlphaFold is not confused by bad decoys. And we use therefore AlphaFold composite score following Rony and Ovchinnikov. However, they used a bit different composite score, but the crucial parameter is the same. So we multiplied the predicted TM score of AlphaFold by TM score between model and decoy. And this term reflects can be interpreted as the measure of how much AlphaFold relied on the decoy, to what extent AlphaFold used the decoy. And after such normalization, we see that Pearson correlation coefficient becomes 86%. And so, if using composite score, we see that AlphaFold outperforms Muster Z-score. And so supporting the conclusions of Rony and Ovchinnikov in their paper. However, and if to compare with previous study of Rony and Ovchinnikov, we followed their implementation of no MSA, no template AlphaFold, but the differences are as follows. We use novel fold structures unseen by AlphaFold and corresponding decoys, while Rony and Ovchinnikov, they used Rosetta decoy library, which was known at the moment of training of AlphaFold. And also we used best threader from ITASER, which is the best automatic prediction tool since 26. And they used Rosetta and deep AtsNet predictors. And so coming back to AlphaFold composite score, we And to this experiment, we can see that still we cannot state that the success of AlphaFold is due to better understanding of physics. Okay, AlphaFold better ranks the decoy structures. However, the setup of the experiment does not allow to prove that AlphaFold success is due to better understanding of physics. Actually, the setup of this computer experiment can only be suitable only to prove that AlphaFold does not understand physics. For example, if in any combination composite score, for example, shows worse performance than performance of Muster. So, and at that point, we come to the ranking of closed structures. Actually, after the paper of Rony and Ovchinnikov, we actually understood that now we have already done the research which can be interpreted as ranking closed structures. And the research was like that. We actually tried to repurpose AlphaFold for some other structural bioinformatics tasks. And one of the tasks is prediction of change of protein stability on mutation. And this task is important for protein engineering and for current, on the current data, the performance of all tools is kind of the order of 50 or maximum 60% of Pearson correlation. Here you see the characteristic plot given produced by FoldX. And when our hypothesis was that actually the only metrics that is given by AlphaFold is the confidence in its prediction. And our logic was like that. If the native structure is native because it is stable, and if AlphaFold's confidence becomes worse, then it could mean that this whole structure becomes less stable. And we correlated the change of the AlphaFold's confidence metrics on mutation and correlated with a change of protein stability on mutation. And we found that actually there is no correlation. Actually, of course, it statistically significantly differs from zero. However, it is too weak to be used for prediction. You see the cloud, and you see the cloud. It is the cloud if to average the confidence over all residues in the protein. And so this means that AlphaFold, first of all, cannot predict delta G. And also AlphaFold success is not due to better understanding of physics, because it cannot rank closed structures by their energy of folding. And so by this experiment, we actually rule out the factor of multiple sequence alignment, and so we see it clear that AlphaFold does not understand physics. Thank you very much, and here are acknowledgements, and I would like to thank Marina, who is a co-author of and main driver force of this project. And please visit our poster, which presented by Marina. It is devoted to prediction of impact of mutation on protein stability using new huge data mega data set published in December last year. Thank you very much for your attention.